Good morning, friends. Welcome back to my YouTube channel Medical Classes by Dr. Srinidhi Kumar Acharya. Don't forget to subscribe my channel and please give your valuable comments. Your comments will help to improve my presentation. And anyway, welcome back to another class of basics of genetics. In the last class, we were discussing about the autosomal dominant trait, and we have also seen that how these autosomal dominant disorders, so they will manifest. And we have seen the concept of penetrance, high penetrance and low penetrance. When there is low penetrance, there is no presentation of the disease. When there is a high presentation or high penetrance, there will be presentation of the disease. To explain the same, one more example is familial hypercholesterolemia. Okay, so first we'll look at this particular pedigree here. And before that, we know something about familial hypercholesterolemia. Now, familial hypercholesterolemia is also one of the autosomal dominant disorder. Okay, this is also one of the autosomal dominant disorders, and this also shows the principles of a penetrance, high penetrance and low penetrance. What I mean to say, first we will have some basics about this disease so that the things will be very clearly understood. Now, familial hypercholesterolemia is one of the example of autosomal dominant disorder. Actually, what happens is there is a gene which makes the low density lipoprotein. Okay, there is a gene present in one particular chromosome. We will not go to that detail. That gene makes some of the structure. What is a structure? Structure is a receptor. Some receptors. Receptor is also a protein, that is also a structure present in the cell membrane. Now, the gene, one gene is there which actually produce the receptors for low density lipoprotein, low density lipoproteins, which is present in the hepatocytes. In the hepatocytes, there is a gene. In the hepatocyte, there is a receptor which is called as low density lipoprotein receptors. This is also called as LDL receptors. And these receptors are made by one particular gene. Now, what is the problem is the gene which is making this LDL receptor is mutant. And as a result, LDL receptors are not made in the cell membrane of the hepatocytes. Okay. Now what happens? Usually the low density lipoproteins contains high cholesterol. We know that. Okay. And these are captured by these LDL receptors. Now every hepatocyte contains these LDL receptors, low density lipoprotein receptors. Now, low density lipoproteins which are circulating in the blood, so that will be captured by these receptors and will come and bind to these receptors. And later, this, after binding to low density lipoprotein or LDL receptors, these low density lipoproteins are internalized into the liver or internalized into the hepatocytes. What's the point? What is the use of these? The use of this is, we know that low density lipoproteins, they contain high amount of cholesterol. So once these low density lipoproteins, so they are get internalized, low density lipoprotein internalized by these LDL receptors into liver. Now liver come to know that how much cholesterol is there in the blood, because this low density lipoprotein is coming from the blood, circulating in the blood. So liver had got a now fair idea that what is the amount of cholesterol present in the circulating blood. So that will be given by this internalized low density lipoprotein. Got the point? Now what happens in this disease is, as the low density lipoprotein receptors are not completely formed or not at all formed because of the problem in the gene which produces LDL receptor, liver hepatocytes do not contain the LDL receptors. So therefore, liver hepatocytes, they are not able to bind with the low density lipoprotein which is coming from the blood. And when they are not binding, the liver is now not knowing exactly what is the amount of cholesterol present in the blood, because there is no receptors, there is no binding. So what liver think now? Now liver think that as there is no lipoproteins are coming inside, so that means that there is no lipoproteins in the blood and there is no cholesterol in the blood. Now what liver will do? Liver thinks that okay, there is shortage of cholesterol in the blood, therefore it is not coming inside my hepatocyte. You don't know that this particular LDL receptor is absent. So it thinks that there is very less cholesterol mostly in the blood, therefore it is not coming to me, LDL is not coming to me, so I have to produce more and more cholesterol. So thinking so, now liver start producing more and more cholesterol. Actually problem is the LDL receptor. LDL receptors are absent or they are less. Now already in the blood also there is more LDL is there, low density lipoproteins are there, and here now liver also wrongly think, okay, confused or wrongly assess that there is no cholesterol in the blood. Again liver start producing more and more cholesterol. No, totally blood is full of cholesterol. Totally blood is full of cholesterol, and that person will have a high amount of cholesterol which start to deposit in the different parts of the body, and that leads to familial hypercholesterolemia. Okay, so this is the problem, familial hypercholesterolemia. Now this usually a familial hypercholesterolemia is a autosomal dominant condition. Dominant means many of the people of the family will be get affected. It do not skip the generation also. Every generation there will be a problem. So this is what exactly happening in case of a familial hypercholesterolemia. Okay, so now we look into this particular pedigree. The same as that of the Marfan syndrome. Okay, here if you see the male is normal and female is having familial cholesterolemia, hypercholesterolemia. Okay, she has got the mutant gene here. LDL receptors are not formed in her body, and as a result it is not able to fix with the low density lipoproteins. Now if they have got in the second generation, they have got 4 kids, the rule is same, 50% males are affected, 50% females are affected. Yes, okay, 50% male and female ratio is equal. In the disease also male and female ratio is equal, male and female. Okay, now what happens? Same. Now this particular affected male, now he marries to a normal female. Okay, normal female. So what happens? Again they will have the 4 kids. The rules are 50%, 50%, disease 50%, 50%, same. Yes, these two have disease, and there is no skipping of generation. Here also there is a disease, here also there is a disease, here also there is a disease. No skipping of generation, vertical transmission. Now suppose you take this particular female who is looking normal, who is looking phenotypically normal. Now she is married to a normal. She is married to a normal male. She is now married to a normal male. Now what happens? This is a normal female. Phenotypically, in the Marfan also we have seen such case, and this is normal female, male, normal female. When they marry, what their surprise again, they have got one child get affected, because this particular female is from the disease family itself. Although disease is not manifested in her body, she has got the tendency. The disease is not manifested in her body because of low penetrance. Okay, but in the next generation, because of so many factors, factorial factors, lifestyle problems, or so many problems, this child developed the disease. And this is again shows the penetrance. This again shows the concept of a penetrance. So we have taken 2 examples for understanding the penetrance principle. Low penetrance may be there. So this mother has got low penetrance. Although she is from the disease family, she didn't have the disease because penetrance is less. She's, in other words to say, she is carrying it. She is in a carrier-like situation. But when it comes to the next generation, although the father is normal, mother is also looking normal, not genotypically, looking normal, the child had the disease, because one gene is sufficient to cause the disease because it's a dominant trait. So this is how the disease familial hypercholesterolemia starts. This is one of the best example for autosomal dominant trait, and also one of the example for penetrance. Okay, so this is another important. So with this we conclude this class. In the next class again we will meet with another important principle of a dominant trait, that is pleiotrophy. So please don't forget to subscribe my channel, and also please give your valuable comments. Thank you very much.